What is gene therapy?
Is it like when you need help picking out genes?
What is gene therapy?
Future medicine.
What is gene therapy?
That is when you use genes to help people that have a certain disease.
Gene therapy is the altering of genes to treat or prevent disease. It is used to treat many genetic diseases such as cancer, hemophilia, and Parkinson's disease, just to name a few. Gene therapy is performed by using modified viruses to carry a healthy version of a gene into a cell. The first kind of gene therapy involves directly injecting the viruses into the patient. Other kinds of gene therapy involve removing cells from a patient's body, inserting the new gene into the cells, and then re-injecting them. The second type works better for white blood cells and could help with immune diseases. The first type, using viruses, could work better for more pleiotropic genetic disorders. There are two different types of gene therapy: somatic gene therapy and germline gene therapy. Somatic gene therapy involves changing the body cells of an individual. This change is confined to this individual and does not affect their future offspring. On the other hand, germline therapy involves changing the gametes or sex cells of an individual, which does affect that individual's future offspring. Currently, germline gene therapy is prohibited by the FDA and research is very limited. Somatic gene therapy, on the other hand, has had some success and could potentially become a widely used technique. The FDA has not approved any of these drugs, but dozens are in the middle of clinical trials.
To better understand gene therapy, we need to go back to 1985. Three trailblazing doctors named Culver, Blaze, and Anderson developed gene therapy by using a retrovirus to carry healthy genes to an infected cell culture. In 1990, a 4-year-old girl named Ashanthi with adenosine deaminase deficiency, also known as ADA-SCID, became the first person to be treated with gene therapy. This is a form of severe combined immunodeficiency which affects the immune system by making the body more susceptible to infections. To begin the gene therapy process, doctors removed some of her blood cells and inserted normal genes into them that could make adenosine deaminase. After the insertion of the normal genes into the white blood cells, the doctors injected her new cells back into her body and cured her of the disease after 2 years of treatments. How do you think gene therapy works?
Like, I don't know, someone consults you about your genes or something? Like,
I don't know. Maybe they I have no idea. I don't know. How do I think it works? I don't know. Something in a lab, I'm assuming. But I wouldn't know how they fix someone's genes.
How do you think it works?
I have no fucking idea.
How do you think gene therapy works?
I guess, I don't know. You just sort of introduce it to the gene, so you add it onto the strand, or like you insert it when DNA is like replicating, or sometime in that process, and then that's when it takes over.
As we learned earlier, viruses are used as vectors to infect target cells with the new gene. If successful, this gene will be incorporated into the target genome, fixing whatever is causing the genetic disorder. This can be accomplished by replacing the faulty genes, fixing mutated genes, or adding a new gene entirely. Genes are often replaced by using viruses as vectors to carry a gene into a cell and having the virus incorporate the gene into the target cell's genome, replacing the mutated gene. For example, replacing a mutated p53 gene could thwart certain types of cancer. The p53 gene works to prevent cancer by regulating cell growth, initiating cell death, and activating repair proteins. If scientists could replace a bad p53 gene, then certain types of cancer that rely on the mutation of this gene could be cured. Turning off a mutated gene that causes disease, such as the dominant allele that causes Huntington's disease, could decrease the severity of certain genetic diseases or even cure the disease. Alternatively, healthy genes could be duplicated so that they are expressed more strongly to promote resistance to disease. Sometimes the immune system doesn't attack diseased cells because they can't identify them as diseased. Use of gene therapy to help the body's immune system identify disease cells as intruders, just like they did with Ashanthi. What do you think can be treated with gene therapy?
Diseases and maybe like problems that like could develop later on in life if they're found early. Like that sequence of DNA is found within the body, it can be replaced.
What kind of diseases can you treat with gene therapy?
Genetic diseases.
Several diseases have a lot of research being done on the viability of gene therapy as a treatment option. We will highlight the ones we find most successful and pertinent to this class. As mentioned previously, immune deficiency disorders was the first successful gene therapy treatment in 1990. SCID and ADA deficiency are the most commonly treated. In these circumstances, stem cells are removed from the blood and retroviruses deliver functioning genes to the patient's defective ones. The stem cells are then reinserted into the patient's body and the virus only infects the target cells. Gene therapy treatments are being created for several types of hereditary blindness, most of which are degenerative. Preliminary trials in animal models have showed some promising results. The eye also allows easy access and a somewhat protected system from the immune system. The viruses which would infect the eye also can't move to other parts of the body. A small clinical trial with patients with Leber's disease shows some improved vision for a few years. Unfortunately, the treatment did not stop the retina from degenerating. There are several gene therapies being developed for various types of cancer, which are showing some promising results. One example is using the herpes simplex 1 virus, which normally causes cold sores. The virus is modified to not cause cold sores, only target cancer cells, and trigger an immune response which would help the body recognize and fight cancerous cells. This therapy, called TVEC, has been used in patients with melanoma and that has spread throughout the body. The viruses are injected directly into the tumors where they replicate, lyse, and infect more cancerous cells. Another approach was taken when treating patients with leukemia. In this clinical trial, patients' immune cells were removed and treated with a virus that caused cells to recognize a protein on the surface of cancer cells. Out of the 59 clinical trial patients, 26 experienced remission.
What are some risks to gene therapy?
No clue.
What would you say is a risk to altering genes?
It could affect your children and grandchildren.
What would you say is a risk to altering genes?
We don't really understand genes super well, I think. Like what changing certain things, like how the side effects it could have. So I guess like the risk in that way, like damaging people or creating proteins, something that we don't understand.
There are many things that can go wrong with gene therapy. If you think about it, scientists are editing information that makes everything in your body work. It's almost like trying to defuse a bomb. You're either going to save someone or detonate something else, like cancer. The first risk is unwanted immune system reaction. Gene therapy is usually accomplished by using viruses as vectors to incorporate new, healthy DNA and disease cells. Because of this, an immune system could identify the viruses as intruders and initiate an immune response. Another risk is all about location. Genes can be inserted into the wrong region on the genome. This can have a similar effect as a reciprocal translocation would. A gene being placed beside a proto-oncogene when it should have been placed by a tumor suppressor gene. The problem with these position effects is that a proto-oncogene and a tumor suppressor gene have opposite functions. The proto-oncogene is like a gas pedal and the tumor suppressor genes are like brakes. This situation led to 5 out of 30 SCID patients developing leukemia after gene therapy. The gene therapy could also target the wrong cell altogether. This can damage already healthy cells and cause problems like cancer. Lastly, the viruses could end up causing an infection in a patient being treated. Most of the viruses are altered so they lose the ability to infect the host, but it's possible the virus could regain this ability.
Who do you think regulates gene therapy?
The government, of course.
I don't really know. I mean, I assume right now it's
private. Just because I haven't really heard much about the government doing anything about it.
Many government agencies are responsible for regulating different aspects of gene therapy. The FDA regulates research and products that are related to gene therapy. The NIH makes guidelines for clinical trials. The NIH Recombinant DNA Advisory Committee was created in response to the public concerns regarding the safety of manipulating genetic material through the use of recombinant DNA techniques. Its purpose is to review trials paying particular attention to ethical, legal, and social issues. The Institutional Review Board must approve any clinical trials before they begin. This is to ensure that the trials are being run properly.
This technology has the potential to completely change our species' genome, so as you can imagine there are some controversies on how and to what extent this technology should be used. One issue is defining good and bad uses for this technology. Should people be allowed to prevent themselves from carrying a gene? Should people be allowed to design their babies? Should this technology only be used on somatic cells? What is going too far? These are all important questions scientists must ask themselves when studying gene therapy.
What do you think of designer babies?
I think it's cool, but I think it can be taken too far. I think the thought of it is interesting, but actually executing it could lead to social issues.
What do you think of designer babies?
The future.
Designer babies are one of the controversies brewing within the gene therapy world. Designer babies are babies that have been modified using gene therapy so that they have certain traits. This can include curing a baby of disease such as hemophilia or cystic fibrosis, but it can also include changing hair color, eye color, or any number of purely cosmetic traits. While it is definitely a good thing to prevent a child from having genetic disease, at what point does tampering become a bad thing? Should a parent be allowed to ensure that their child grows up to be tall and athletic? Another issue within gene therapy is cost and who will pay for the treatment. Gene therapy costs over $1 million. Should insurance companies pay for costs associated with gene therapy? If insurance companies don't pay for gene therapy, the cost will have to be covered by families. This would likely lead to many families being unable to afford gene therapy. While gene therapy can definitely be used to improve many people's lives, there are a whole host of controversies associated with it that must be examined and resolved before gene therapy can be used to its full potential. This leads us into the future of gene therapy and how it may eventually be used or not used.
Although the prospects of gene therapy are great, many will wonder why it isn't being implemented all over the world. Due to the potential risks, more technological advancements must be made before gene therapy is introduced as a typical form of treatment for disease. Gene therapy has the potential to correct genetic diseases, eliminate cancer cells, decrease the amount of patient visits to the doctor by personalizing medicines, and much more. Overall, it could substantially increase the quality of life for someone. As the technology advances and more research is done on gene therapy, the risks will shrink and the therapy will be more reliable. The first car Henry Ford invented was not as safe and efficient as the cars in the world today. It took years of testing and development to get it where it is now. This is the same for gene therapy. It will take time to advance the technology and become more reliable, but once it does, the medical world will forever be changed.